Today we're jumping into a real scientific detective story, how we cracked the code and figured out the structure of DNA. Let's get right into it.
I mean, have you ever really thought about that? How can one microscopic molecule have all the instructions to build, say, a giant redwood tree, or a blue whale, or you? The secret, it turns out, isn't just what it's made of, but its absolutely incredible shape.
So first, the basics. DNA, or deoxyribonucleic acid, if you want to be formal about it, is essentially the software of life. It's running in almost every single one of your cells, dictating pretty much everything. But, to really get how it works, we first have to understand what it looks like. And our story doesn't start with some crystal clear image. Nope, it begins with a fuzzy, mysterious shadow, the very first clue that would end up changing biology forever. This first critical piece of evidence came from the work of a brilliant chemist, Rosalind Franklin. She was using this amazing technique called X-ray crystallography, which, for the very first time, gave scientists a peek at the actual structure of the DNA molecule itself.
So how on earth does that work? Well, it's kind of like you're in a pitch black room and you're trying to figure out the shape of a complex chandelier just by throwing tennis balls at it and mapping where they bounce off. You fire X-rays at DNA, the rays scatter off the atoms, and they create this pattern of spots on a piece of film. That pattern is basically a map of the molecule's guts. And this was the aha moment. The image she captured, famously known as Photo 51, had this unmistakable X shape right in the middle. Now, to a crystallographer, that cross pattern is a dead giveaway. It basically shouts that the molecule isn't a straight line, it has to be a helix, a spiral. But that shadow on the film was so much more than just a shape. Hidden inside that pattern of spots was a ruler, one that gave these incredibly precise measurements that would become the absolute foundation of the final model.
Yeah, you heard that right, a ruler. The spacing between those dark spots on the film could be translated directly into physical distances inside the molecule. It was mind-blowing. And the unit of measurement here was the angstrom, which is ridiculously small, just 1/10 billionth of a meter. They were literally reading a story told by shadows.
So, the first number they pulled from the photo was 3.4 angstroms. If you imagine DNA as a spiral staircase, this number represents the vertical distance from one step to the very next one, the height of a single rung on the ladder. The next clue was a simple number, 10. The pattern clearly showed that it took exactly 10 of these steps, or 10 base pairs, to make one full 360-degree twist of the spiral. And this is where the visual evidence just turns into simple, beautiful math. If each step is 3.4 angstroms high, and you've got 10 steps in one complete turn, well then the total length of that twist has to be 34 angstroms. It just fits.
Okay, but the final measurement was a bit of a head-scratcher. The data revealed that the DNA molecule had a perfectly consistent width, or diameter, of 20 angstroms. It didn't get wider, it didn't get narrower, it was perfect. And that raised a really big question.
I mean, think about it. We knew DNA was built from 4 different chemical blocks, A, G, C, and T. Now, if these blocks were different sizes, wouldn't the latter kind of bulge in some places and get skinny in others? How could the width be so perfect?
That question was the last big piece of the puzzle. Answering it meant someone had to take all the clues, the spiral shape, those exact measurements, and the chemistry, and somehow put them all together into a single working model. And this is it, the iconic double helix. This is the model that James Watson and Francis Crick proposed in 1953, using Rosalind Franklin's absolutely crucial data. It was elegant, it was simple, and it perfectly explained every single piece of evidence they had. At its heart, the model is really just a twisted ladder. You have 2 long rails on the outside made of sugar and phosphate. Fun fact, they actually run in opposite directions, what scientists call anti-parallel. And then the rungs connecting them are the bases, all stacked neatly and safely on the inside. So back to that constant width problem. How did they solve it? Well, it turns out the bases come in 2 sizes. You've got your bigger ones, the purines, adenine and guanine. They have a 2-ring structure. And then you have your smaller ones, the pyrimidines, thymine and cytosine, which only have a single ring. And here is the absolute genius of the whole thing. There's an unbreakable rule. A big purine on one side
always has to pair with a small pyrimidine on the other. No exceptions. Adenine always pairs with thymine, and guanine always pairs with cytosine. That's it.
And there's our answer. A big piece plus a small piece always equals the exact same total width. This simple rule makes sure every single rung on that ladder is the same length, which is what keeps the whole helix a perfect constant 20 angstroms wide. It's an incredibly simple and elegant solution to a big puzzle.
But look, this structure isn't just beautiful to look at, it is profoundly functional. The specific shape of the double helix is the secret to how life itself manages to continue on and make copies of itself. Because the strands are what we call complementary. If you know the sequence of bases on one side, you automatically know the sequence on the other. This lets the molecule unzip right down the middle, and each half serves as a perfect template to build a new, identical partner. This is it. This is how life replicates.
So this design is it's genius. You get redundancy, right? The information is stored twice, which is amazing for proofreading and repairing damage. It gives you a super simple way to replicate. And it provides stability by tucking all the important genetic code on the inside, safe from harm. The form and the function are just perfectly intertwined.
When you think about it, the structure of DNA shows us how unbelievable complexity can come from just a couple of very simple, elegant rules. A pairs with T, G pairs with C, and from that simple foundation, the entire breathtaking diversity of life on Earth is built. It really makes you wonder what other profound truths about our universe are hiding in plain sight, just waiting for us to decode them.